Thank you very much to allow me to do this presentation today, because it's kind of special for me, as my first presentation as a PhD student was at the EvolTree First Conference in 2020, and the last one will be today, so thank you again to allow me to present today. So now I will talk to you about the genetic architecture of leaf specials metabolites not through ovation in sessile provenances. So a very brief introduction about white oaks. So first of all, white oaks are a dominant tree species in Europe and a dominant tree species in European temperate forests. And in our study, we are studying more specifically sessile oaks, and sessile oaks represent 15% of French forests. And in our study, we are studying the specialized metabolites. So a very brief definition of specialized metabolites. It is chemical compounds that are not directly involved in plant development. However, specialized metabolites play a key role in plant defenses. And leaf metabolites in oaks were previously studied, and multiple studies demonstrated the role of specialized metabolites in herbivory, but also in resistance to abiotic stresses like drought. So in our study, we are interested to identify the genetic basis of leaf specialized metabolites within sessile oak. And we want to answer two questions. So first, is there variation of leaf specialized metabolites among and or within sessile oak provenances? And also, what proportion of the variation we observed in leaf specialized metabolites is explained by genetic variation? So to do so, we sampled leaves from oak trees that grown in a common garden in North France. So here represented by the black triangle on the map. And we sampled 225 trees from 9 different provenances that are along a latitudinal gradient from North France to North Germany. And from each tree, we sample leaves at 2 different branch heights. We sample low leaves that are low branch height that are mostly protected from sunlight. And we also sample high leaves in high branches that are exposed to sunlight. And from these leaves, we perform first whole genome low-depth sequencing using Illumina sequencing, and we obtain 1.4 million genetic markers, and here we are using SNPs. And also, we perform high-throughput untargeted liquid chromatography coupled to mass spectrometry to measure the signal intensity variation of leaf specialized metabolites in sessile oaks, and we obtain signal intensity variation for 217 molecules. So now, if we are interested, we studied the genetic variation of our provenances. So first of all, we calculated a differentiated index, the FST, between all pairs of populations. And we obtained values between 0.01 and lower value, which means that we have very low FST values. That means that we have a weak population structure. And we also perform a principal component analysis using 350,000 markers. And we can see that here, each dot corresponds to a tree colored by the provenance, and we are able to capture the weak population structure with our genetic markers, with here the Vacher and grazing population represented here. So now we observe that we have genetic variation in our provenances. So as I said before, we also perform high-throughput untargeted liquid chromatography to measure signal intensity signals of metabolites. So we perform putative annotations to measure which compounds we obtain from this analysis. And here we obtain this annotation by similarities of chemical structures with plant databases. And we obtain different ontology classes like flavonoids, terpenoids, also chinic acids. And these different compounds were previously studied in oaks, and the different studies observed their role in biotic interactions like in Tortrix viridana or in different pathogens like Phytophthora cinnamoni. So now we studied the variation of these metabolites within oak provenances. So just for the presentation, here I will just present the result from the high branches, but we observe the same pattern in low branches. And here we are removing the height effect of the variation, for the variation of leaf metabolites. So we perform a principal component analysis using the 270 molecules as metabolomic profiles of each tree. So here, each dot corresponds to a tree coded by the provenance. And as you can see, here the different circles represent the 95% confidence levels, and we are not able to group the provenance thanks to their metabolomic profile. And if we are looking at each metabolite here, here on the y-axis we have the pseudomolecular intensity, and each dot corresponds to a tree, and each tree is grouped by the provenance along the y-axis. And we observe that we have a high intrapopulation variation of least species metabolites for most of the molecules we investigated. But we were also interested to find if we have molecules that display differentiation of variation between our populations. So to do so, we also perform a partially least square discriminant analysis. And here, again, each dot corresponds to a tree colored by the provenance of the tree. And this time, we identify differences between the provenances here with the grazing population compared to the buzzing population. However, when we are looking at the percentage of the variation of the first component, we can see that we have less variation, 3%, compared to the participant component analysis of 9%, which means that here the variation we observed didn't explain the majority of the variation we measured for leaf metabolites. And here, if we are looking at the molecule that allow to discriminate the two population, this metabolite, we can see that we have higher intensity values of the grazing population compared to the other population. But we can also see that we have a high intrapopulation variation for the other populations. So we observed a low interpopulation variation, but only for few metabolites we investigated here. So now we observe that we have genetic variation and intrapopulation variation for leaf species metabolites. So we identified the genetic basis underlying this variation using genome-wide association analysis. So we performed genome-wide association analysis for the 297 molecules, and we obtained significant association for 93 of the molecules. Here we have represented 3 different Manhattan plots for 3 different molecules. And here each red dot corresponds to a significant association with genetic markers, and the red circles correspond to the highest SNPs associated with the molecular variation. And also from the 93 molecules and the significant association, we identified 321 Coq candidate genes. So here we have represented the intensity of the molecules explained by the genetic markers. So here, each dot corresponds to a tree, and here, this time, the color corresponds to the genotype at the highest associated marker with the molecular variation. So here, as an example, for this one, the genotype here corresponds to the SNP here. And so we can observe that we have different 2 classes with higher intensity signals associated with heterozygous genotypes, individuals, and that we have also lower intensity signals that is associated with homozygous individuals. And also we observe that markers associated with molecules variation are polymorphic in all 9 provenances. So that raises a new question: how genetic diversity associated with leaf species metabolites is maintained within populations? One hypothesis is that it could evolve naturally. However, it's not. It is not what we are thinking, because leaf species metabolites are involved in plant defenses, and also we observe genetic variation and genetic drift of the populations. So another hypothesis is that species metabolites may have a pleiotropic effect. Here I'm taking an example from a previous study that observed a maintenance of polymorphisms associated with glucosinolate production in Boeckra stricta. In this study, they observed that both herbivory and drought were both drivers of selection on glucosinolates, and that these two, herbivory and drought, selected different alleles, and we have a trade-off of alleles at the candidate gene associated with the glucosinolate production. So here, if we are go back to our study, here the molecule we obtained with a very strong association with genetic variation. We annotated this molecule as a thiogalin, or also called a galloic chinic acid. And from our knowledge, we didn't identify the biological activity of these metabolites in oaks. However, this compound is composed of two moieties, chinic acids and gallic acids. And these two moieties were previously studied in oaks, and different studies observed that these two moieties were involved in different processes in response, for example, for chinic acids to wounding, water deprivation, temperature elevation, and also gallic acid involved in herbivore resistance, but also they observe seasonal variation in response to temperature differences between seasons. So here it is possible that these metabolites may have a pleiotropic effect that will select different alleles with the combinations of the different stresses. And also another point that I won't develop here today, but here we obtain a serine carboxypeptidase-like as a candidate gene associated with the genetic marker here. And this candidate gene is known to be involved in special metabolism pathways in different plants like grapevine. And also, we observed that this candidate gene is a tandem duplicated gene. And what is very interesting is that in the previous study I presented here, they also observed that the candidate gene associated with the glucosinolate production is a duplicated gene. So I think we have something to investigate it here to investigate the potential role of duplication in specialized metabolism variation. So to conclude, we have two main questions, and we answer so we observe that we have low phenotypic differentiation among provenances, and that most of our variation is within oak provenances. We identified a separated architecture, mono to oligogenic architecture of the variation, and also we observed that polymorphisms are distributed within the 9 provenances. So that raised the new question, how genetic diversity associated with leaf specialized metabolites is maintained within populations. So today I presented one hypothesis, a pleiotropic effect of metabolites, but another hypothesis is maybe a microlocal adaptation, which means that we have adaptation at a finer scale than at the population level. But if you want to discuss about this hypothesis later, we can do it. So first of all, thanks to all the collaborators and co-authors of this work, and also the fundings that allow me to do my PhD. And thank you for listening, and I will be very happy to take questions.
Yes, thank you very much, Domitille, for a very special talk and very great talk. I'm excited if there are any questions. I guess it's a very special thing. Okay, there's a question over there. Yeah.
Thank you for the presentation, and I really appreciate all of the analysis because I know how hard is the HPLC analysis or to interpret the results.
Yeah.
I would like to know if you try to do this similar analysis with something more easy, like a chlorophyll content, dry matter content, water content, or something like this, which is easy to collect and easy to interpret.
Yeah, unfortunately not. We just measured in our common garden experiment and done a screenshot. Yeah, we only do that. But for the future, we want to study the the role of the candidate gene we obtained in herbivory experiment or drought and measured and sequenced genotypes and measured also metabolomics and try to study if we are able to see the variation of metabolites linked to the genotypes we identified with herbivory experiments. Yeah, thanks.
Okay, there's another question up there. No, sorry, yeah, yes, but first, yeah, over there, please. You can give him already the microphone.
Hi, thanks very much for this fascinating presentation. You mentioned that the patterns that you saw were very similar regardless of whether you were looking at the samples taken from the top of canopy or the samples taken from the bottom of canopy. Yeah. And I was wondering if you could expand on that a bit. I was a little surprised. I could imagine that within that the diversity levels might be similar in these two groups, but I would think that you would find some systematic differences in the metabolite profile between top of canopy and bottom of canopy leaves.
Yeah, I didn't present it here today, but in fact we observed, we performed a PCA to study the variation of high and low branches, and we are able to have the two groups. So we have differences of between the high leaves and the low leaves. However, we also measured that this variation is highly correlated. And so we have differences, but in fact, when we perform the analysis of population differences from high branches and low branches separately, we observed the same pattern, as they are highly correlated. And also what I didn't mention here is that I performed genome-wide association with the two branch heights separately, and we obtained the same candidate SNPs for both branch heights from the same metabolites.
Just going along with the similar question, did you compare, for example, two top branches from the same tree and see how variable the signal is from the crown globally?
It's a very good point. So high branches from the same tree, but not located at the same position of the canopy. That's right. Yeah, I didn't look at it, but I think it'd be very interesting, and we have the data to study that, to see if we have variation at the high. Yeah, because maybe we can have an effect of like in common gardens, oaks may be very close to each other, and maybe we say that it's exposed to sunlight, but maybe the neighboring tree is hiding a little bit the high branches. So maybe it can be also very interesting to look into it.
My question goes in the same way, because you only use one high leaf per tree?
Yeah.
Because because I'm used to the colleagues from the Helmholtz Institute, and they always use at least 3 technical replicates. You use one. Okay, because of course the variation within a tree is so high. Yeah. Okay.
So here we only use one, but we have replicates. And we corrected with it, but here we removed it for variation analysis.
Okay, so you can check it, yeah?
Yeah,
okay,
we can.
Yeah. Okay, thanks. Kata.
Thank you. I was wondering, thanks a lot for the great talk.
Thanks.
And I was wondering, so do you know anything about the costs that are involved in expressing these secondary metabolites? Because if it's a costly trade, I could imagine in the moment when the herbivore density goes down, then the other allele might be favored. Yeah, I was just wondering.
Yeah, I don't know for these specific compounds, but it may be you have. Plants have a trade-off in producing primary metabolism or specialized metabolism. So, and I think we'll have a presentation talking a little bit about that just after. But here I don't know, for my case, the cost to produce these specific compounds in response to stresses, as I only measure a screenshot of one time point and not during a stress event.
Thanks a lot.
Okay, thank you very much, Domitille.
Thanks.
It's a very important and interesting part. Of course, we already discussed it, and we will discuss it further, of course.
Yeah, thank
you. We now thank you again.